if you look at how disease has been treated for 50 years, for 100 years, it's basically with a relatively narrow range of pharmaceutical compounds with unpredictable results.
We treat a population of people with a certain drug and say, okay, more than 50% of those responded to that drug, so that's a good therapy.
That's a drug for everybody, but it's not.
It wasn't 100%, and it's never 100% because we're all different.
Each of us has a genome that's roughly 3 billion lines of code.
All of us, our genomes are different. So all of us are susceptible to different types of diseases, different types of cancers, different types of therapies and drugs.
The Human Genome Project took 13 years and cost $3 billion to sequence one genome. And now we can sequence a genome overnight.
We now live in a world where we can take the genetic profile of an individual
and use that to tailor treatment of human diseases. That's what genomic medicine's about.
Genomic medicine is personalized medicine.
The hope is to try to find therapies that can actually target this diabetes and that diabetes, this cancer and that cancer.
The question is translating that science into therapies as quickly as possible. The place is that it was set up specifically to undertake genetic research before genetic research was cool.
Clarence Cook Little was a Harvard-trained geneticist who founded the Jackson Laboratory with philanthropic support from Edsel Ford and Roscoe B. Jackson of the Hudson Motor Company.
Good morning, Jackson Laboratory.
Over the past 85 years, we've made a number of incredible discoveries.
If you look, for example, at organ transplantation,
Blood transfusions,
in vitro fertilization,
The discovery of stem cells.
all of these, when you go back in time, have a source in the Jackson Laboratory.
We've contributed to somewhere on the order of 25 Nobel Prizes.
The term genomics originated from the Jackson Laboratory. They defined, actually, genomics back in the 80s. A lot of people don't realize that. Yeah. Google it up. You'll find it.
the mice are what helped us get those techniques and set the stage for those medical advances.
One of the things that we're doing is looking at the birth of the cancer. We start from cell one. So we start from a single stem cell. And we engineer that cell to gain those mutations that we think are driving tumor formation. We have no ability to do that in human beings. But we can do this in human cells that we put into mice. That is such a powerful system to understand what's driving that cancer and creating it from a healthy cell.
The mouse has between 95 and 98% of all the genes that we have. Think about that. It is a model of the human being.
Because we had, over decades, built this incredible repository of information on mouse genomics. We're able to take our insights that we have learned over the past 85 years and bring it to real patients in the real world.
I was speaking with a clinician a few weeks ago. I asked him, if there was one piece of information that would change the way that you treat patients every day, what would it be? And he said, if I could know which one of those people are going to eventually develop leukemia, it would change the way I treat them today.
They told me I had CLL, and CLL. That's just 3 letters. But you take the letters and then you spell out the name, then you see that word, which is, a cancerous name. When I hear the word cancer, I kept saying to myself, I hope that's not me, I hope that's not me. But when that day came, I said, yep, now it's me. It's scary in a way because when they wipe out your immune system, you can catch, the snap of your finger, anything that's going around. I mean anything. So you got to be really cautious.
One of the defining features of CLL is its inability to faithfully and accurately copy its genes. CLL cells, like so many other cancers, make mistakes. Because of that, cancer cells become critically addicted to DNA repair. If we can take away some of their DNA repair capacity, we can actually cause a CLL cell to essentially mutate itself to death. Right now, we measure 5-year survival rates. If we can succeed, we can begin to think about 10 or 15 or 25-year survival rates, so that Al's story becomes a thing of the past in cancer therapy.
If you were unfortunate enough to be diagnosed with cancer today, you would have less than a 35% chance of getting the right chemotherapy the first time.
The historical approach to treating cancer is kind of a one-size-fits-all treatment. A treatment approach that's no different from one cancer patient to the next. Now that we're in the genome era, we can begin to think about a different way of treating cancer.
So rather than, you have brain cancer or you have lung cancer, you have a cancer that's caused by this mutation. And we have a drug that targets that mutation.
A mouse avatar is really a group of mice who represent us.
That mouse becomes your surrogate, your avatar in the experimental space.
We start with a little piece of tumor from the patient and we put it into one mouse. And it grows and we put it into 5 mice. Then we take the tumors out of those 5 mice and we put it into 5 more mice.
It's almost like a photocopy process, right? You have many mice now all carrying a tumor from an individual patient, and you start treating those mice as if they were the patient.
So we can try drug A, B, and C. We can try A and B in combination.
And you say, aha, treatment A works, treatment B doesn't work, right? And you do that testing in the mice, the tumor-bearing mice, instead of on the patient.
So now we have the ability to actually test new drugs and new ideas in a matter of months, not in a matter of years.
The real value in the avatar is the ability to look deeper into that cancer, to figure out something bigger and broader that can be applied to millions of cancer patients.
The Jackson Laboratory is accelerating the pace of discovery.
The edge that JAX has is the ability to work directly with human disease, put it immediately into the mouse, scale it up, test it, share it, and get it back to benefiting people. No one can do that as fast as we can, and no one can do that on the scale that we can.
We're dealing with the biggest and most challenging problems known to man. The thing we're short on are the resources to take and translate those discoveries and insights into therapies.
When I came to the Jackson Lab 20 years ago, I could not have predicted that I would be working on research that I think in my lifetime is going to have a positive impact on the way we think about and do patient healthcare.
But it takes my support and it takes your support for the purpose that motivates us, which is the science, the cure, the patient, helping human beings.